Okay, so for 7-2, you have a cross between individuals with genotypes big C, little c, big D, big E, little e, and homozygous recessive for C, D, and E. They have 1,000 individuals, individual offspring. The class that was big C dash or big C dash, big D dash, little e had 351 individuals. And so now it's asking if any of these any of these genes are located on the same chromosome or linked. Okay, and so what you have to do is you have to think about, okay, if this was just our typical trihybrid cross, test cross actually. So first you have to recognize that it's a test cross because you're crossing an individual that is well, we know the genotype in this case, but if we didn't know the genotype, it would be suspected heterozygous in each of those individual genes with a homozygous recessive at each of those genes. And if we did that kind of cross, we would expect a ratio. If you remember, with our test crosses, it's always the 1-to-1 kind of ratios. So in a trihybrid cross, you would have 8 different phenotypic classes, and then you would expect a equal representation in each of those different classes. So you expect a ratio of 1-to-1-to-1-to-1-to-1-to-1-to-1. Okay, and so that would essentially be 1,000 of the 1,000 individuals. You divide it by 8, and you would expect down here 125 individuals in each of these classes, right? What we see is that in one of the phenotypic classes, you actually have 351. So you know this is a big deviation, right? So the best answer, or the best way to think about this is that, okay, probably at least 2 of the genes are linked, and that's why you're getting these deviations from the 1-to-1-to-1-to-1-to-1-to-1-to-1 ratio. Okay, and that's all that problem is asking you. You just need to know, you need to realize what you would expect in each of these classes, and you would see that in fact um, you have deviations from what you expect, probably based on the big C and big D being linked or closely linked to each other, therefore giving you an overrepresentation in that class.